Hi everyone, welcome back to my A-level biology lecture series. In this lesson, we will explore chemiosmosis in chloroplast. At the end of this lesson, you will be able to explain that during photophosphorylation, energetic electrons release energy as they pass through the electron transport chain. The released energy is used to transfer protons across the thylakoid membrane, and proton return to the chloroplast stroma from the thylakoid space by vascular diffusion through ATP synthase, providing energy for ATP synthesis. Let's get started. Chemiosmosis in chloroplast. How this reaction take place inside the chloroplast. Focus on this diagram. Between photosystem 2 and photosystem 1, there are some electron carrier which form electron transport chain. During photophosphorylation, electron from the primary electron acceptor of photosystem 2 is transferred along this electron transport chain and finally received by the reaction center of photosystem 1, which is P700. During this process, electron releases energy, and this energy is used to transport protons from chloroplast stroma into the thylakoid space, again its concentration gradient. Therefore, there is an accumulation of proton inside thylakoid space. Besides this, you can see that during photolysis of water, protons are produced. So therefore, with these two events, the concentration inside thylakoid space is very high. However, proton concentration inside stroma is low. A steep proton concentration gradient is created. And this proton concentration gradient can be termed as electrochemical gradient. However, proton can't pass through the phospholipid bilayer that form thylakoid membrane. The only way for protons to move from thylakoid space back to the chloroplast stroma, it must pass through proton channel, which associated with ATP synthase. So when proton move from thylakoid space back to the chloroplast stroma by facilitated diffusion, energy is released. So this energy is used by ADP and inorganic phosphate group to form ATP, and this reaction is catalyzed by ATP synthase. And these protons will combine with electrons, okay, together with NADP to form reduced NADP. So both reduced NADP and ATP are required by Kelvin cycle. That is how chemiosmosis actually takes place in chloroplast. So in order to achieve this process, first step is very important to establish proton gradients in the thylakoid space. So as electron move through a series of electron carrier, energy is released. And this energy is used to pump proton from chloroplast stroma into the thylakoid space against its concentration gradient. So the proton concentration in the thylakoid space increases further due to the proton release from photolysis of water. The next step, which involves ATP synthase in or during ATP synthesis by chemiosmosis. So the thylakoid membrane is impermeable to proton, so they can only return to the chloroplast stroma by vascular diffusion through a proton channel, which is associated with ATP synthase. As protons flow down their electrochemical gradient, energy is released, and this energy is used by ATP synthase to catalyze the phosphorylations of ADP to form ATP. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.